Covering the unit of digestion, which is unit 6 of IB core biology content. So in this video, we will talk about the anatomy of the digestive system along with the different functions of different organs, the two different types of digestion, the involvement of enzymes in the digestion process, structures specifically of the small intestine and how nutrients from the food is transported into the small intestine, into the bloodstream. An example of starch digestion will be looked at in detail. The role of the pancreas will also be discussed, and we'll be finishing the video off with modeling digestion. So beginning with the anatomy of the digestive system, we can see that it is consisted of the alimentary canal and other accessory organs, which I labeled in red and blue. So the alimentary canal is essentially consisting of organs where food actually passes through. So that includes the esophagus, the stomach, the small and large intestine, including the rectum. Whereas the accessory organs, they are consisted of organs that actually aid in digestion, but don't actually transfer or involve the passage of food. And that includes the salivary glands, the liver, pancreas, gallbladder, and we'll begin by discussing the salivary glands, which is where saliva is released, and this is located somewhere in the mouth, and that's where the first stage of starch breakdown or digestion actually begins, and that is because it includes the enzyme salivary amylase, and we know that amylase breaks down starch. And also, yes, the saliva also moistens food, which makes the passage of the bolus of food easier as it passes down the esophagus, which leads us to the next organ, which is the esophagus, and that's basically connecting a hollow tube that connects between the mouth and the stomach. Next, the stomach is basically a container that mixes food by a process called churning, and that mixes stomach acid, which is secreted by the gastric pits lining the stomach, with the food particles, and that that's where like protein digestion happens because it has a suitable pH range of 1 to 2, and it's like very acidic. And that's where protein digestion occurs. So moving on to the liver, that's where bile production happens. It also has a role of detoxification. And the gallbladder is where the bile produced by the liver is stored. And the pancreas has a really important role of not only secreting enzymes, but also regulating blood glucose levels through hormone productions, and these hormones will include these hormones are glucagon and insulin, and that will be discussed in more detail later on in the video. So the small intestine is mainly where nutrient absorption happens, and it's divided into 3 sections: the duodenum, and I don't think we really need to know this one, but like I feel like this is the main part that we actually learn about in IB biology. So in this section, I think when food enters from the stomach to the small intestine, actually that's where bicarbonate ions are released into the small intestines to actually neutralize the stomach acid, to make it more alkaline or neutral, to provide a suitable range of pH for the enzymes in the small intestine. And moving on to the large intestine, that's where most of the water and mineral absorption occurs, and it's consisting of all these sections, which we don't really need to know in detail, as well as the rectum, where feces is stored before it's passed out of the body, the system. So there's 2 types of digestion, and the first one is mechanical digestion, and that's mainly involving the process of chewing, which is basically breaking down larger sized food food pieces into smaller pieces by breaking them down with your teeth. And So yes, there's the epiglottis preventing the bolus food from entering the trachea, and I believe this part we don't really need to know this, but yeah, it's to ensure that the food actually travels down the esophagus. And then we have chemical digestion, where it's basically breaking down large insoluble particles into smaller ones, or its constituent like monomers or monosaccharides that actually gets absorbed by the body. And during this process, three things are involved. We have the stomach acids, the bile, and enzymes. So enzymes are biological catalysts. So these things we might have we were probably familiar with from IGCSE, but enzymes, they're biological catalysts that increase the rate of reaction by lowering the activation energy, and it remains unaltered after each catalysis reaction catalysis. Bile, it emulsifies fats into smaller droplets, and that increases the surface area for the enzymes to act on subsequently. And the stomach acids, they denature the proteins, which aids in protein digestion. So, and then we're also going to talk about the peristalsis, which is basically a movement of food from the esophagus along the alimentary canal in a unidirectional manner. So it only goes in one direction, which is from the mouth to the rectum or the anus. And that involves the contraction or relaxation of longitudinal muscles. And we also have segmentation, which involves circular muscles, and this is a bidirectional movement, and that involves the contraction of non-adjacent circular muscles. It also better mixes the digestive juices with the food. So moving on to different enzymes and their corresponding substrates. So first of all, we have carbohydrates, and that is targeted. So the enzyme that targets carbs is amylase, and you can find them in the mouth. It's also produced by the pancreas. You can find them in the small intestine. It breaks starch or other carbohydrates into the monosaccharides of glucose, fructose, and galactose. And then we have proteins, which breaks down into amino acids, and it has actually different ranges of optimal pH. For example, for protease, for pepsin, its optimum pH is pH 2, whereas this one is, I'm not really going to say, it's kind of like a long word, but it functions best at a pH of 7. So that's near the small intestine, which we know has a relatively alkaline or neutral level of pH. And then we have lipids, which is broken down by lipase, normally in the small intestine, and it breaks lipids down into fatty acids and glycerol. So, well, yeah, and then I believe we haven't really covered this a lot, but nucleic acids, they are broken down into nucleosides. And so enzymes, as we mentioned before, they're biological catalysts. They remain relatively unaltered after breaking down the substrate into its products. So we can imagine this one, for example, to be lipids, and the products here will be fatty acids and glycerols. And during this process, an temporary enzyme substrate complex will be formed. And yeah. So moving on to the structure of the small intestine. So as we briefly talked about before, this is where the nutrient absorption occurs. So if we look at this diagram, there is the lumen, which is basically the empty, it's like a passage where food actually passes through. So this is like a hollow, tubing almost. And then we have a highly folded layer called the mucosa, which is where material absorption happens. It crosses, so the material that's absorbed crosses the epithelium and enters the bloodstream. The next layer we have here is the submucosa. So I think it's this layer here. Where is it labeled? Here, submucosa, and also in this this cross section, we can see it's like around here. So the submucosa acts as a separational barrier between the mucosal layer as well as the muscular layer, and then the muscular layer, as we can see, is over here. So it's also called the muscularis externa. The outer part is the longitudinal muscles, which is responsible for peristalsis, and then we have the inner circular muscles, which is responsible for segmentation. And then this outermost layer, this really thin outermost layer, is known as the serosa, and that's a protective outer layer and also secretes serosus fluids to reduce friction during the movement of the muscle layer. So we can see, so in the small intestine, there are many finger-like projections known as villi, and on these villi, there's more small finger-like projections. As we can see from one of these sections, there are so many microvilli. So the main purpose of the microvilli is actually an adaptation of the villi or small intestine itself to increase the surface area for material absorption, nutrient absorption. It also has a dense capillary network, which increases the rate of nutrient transportation. As we can see in this cross-section diagram, there's a lot of blood vessels. It also has a really thin epithelial layer, which is 1 cell thick, and that's to minimize the distance for the diffusion of the absorbed nutrients and molecules into the bloodstream to the rest of the body. And there's also lacteals that's present, which transports lipids into the lymphatic system. I don't think we can really see that in the diagrams here, but there's that. There's the lacteals. There's also crypts and pits, which releases the digestive juices, as well as membrane proteins, which we can't really see in this in these diagrams, but there's membrane proteins, and we're going to discuss each of their functions and the materials the molecules they transport later in more detail in later slides. There's also the presence of mitochondria, and that's to provide ATP for the active transport, which is one of the membrane transport mechanisms that we will discuss later. Oh, right here. So, active transport. So during active transport in the small intestine, the sodium ions are actually pumped against this concentration gradient. So that's from low to high concentration gradient outside of the membrane, which creates a high concentration of sodium ions outside of the epithelial layer, and that's going to bring back monosaccharides of starch, for example, such as glucose and galactose, as mentioned here, via cotransport, and they're going to diffuse passively back into the cell. And, well, there's also facilitated diffusion, which is this diagram. So that's mainly for, sorry for not mentioning this, but active transport also actively transports molecules such as amino acids and mineral ions. So facilitated diffusion diffuses hydrophilic molecules through the phospholipid bilayer into these epithelial cells. So it's also for monosaccharides, such as fructose, and also the diffusion of vitamins, as we can see listed here. Osmosis is just for like water, as we know, and simple diffusion, we have, for example, lipids. So actually lipids, in this diagram mentions triglycerides, but essentially lipids can travel through simple diffusion because they're able to diffuse passively through the hydrophobic core of the plasma membrane into the epithelial cells, and they'll be carried away via the lacteals. So here in this diagram, we see this vesicle being formed, and this process is called endo or penocytosis. So penocytosis is for like liquids, but yeah, both of them require substances taken in by the plasma membrane, without without digesting them, and that's done through the invagination of these membranes, as we can see. So moving on, starch digestion. So a polysaccharide here, we have starch. So this is just a brief example of starch digestion in the chemical digestion process. So it's going to break starch down into disaccharides such as sucrose, maltose, and lactose. So the composition of sucrose, for example, is one glucose and one fructose. So I think we have to know these specific, the compositions of these disaccharides actually. So the way I was taught to remember what sucrose is composed of is you think of glucose. So there's always going to be a glucose in all three of these disaccharides. And one of them, just think fructose, think of fruits, and that's sweet, so sucrose. If that makes sense. Maltose, you just have 2 times glucose. And for lactose, there's 1 galactose and 1 glucose. So galactose has basically lactose inside, so that's more straightforward to remember. And yeah, so the role of the pancreas. The pancreas secretes enzymes, for example, amylase, and hormones, and that includes insulin and glucagon. So insulin will essentially lower the blood glucose level by converting or storing glucose in the form of glycogen, and yeah, it stores glycogen in the liver as well as adipose tissues. So glucagon will increase the blood glucose level, so it's like the opposite of insulin, sort of like, I think the word's like antagonistically. It increases the blood glucose levels by limiting glycogen synthesis and storage, or it also breaks that down, takes it from the liver and releases it, breaks down glycogen and releases it back to the bloodstream to increase blood glucose level by breaking it down to glucose. And we can see, yeah, that's just the diagram illustrating this mechanism. And here, finally, we are ending with a model for digestion. So the first experiment, that's essentially showing how, so first of all, the experimental setup is we have a control group and we have an experiment group. So the control group consists of a dialysis tubing with starch solution only. So these dialysis tubing kind of model or resemble the small intestine in a sense that it's semi-permeable, and it only allows the passage of certain molecules. For example, we can see in this diagram these large molecules such as starch and things like that. It's too big to pass through these perforations, so it's not going to pass through. And these small molecules or salt ions that are small enough are going to pass through. So in experiment 1, I believe it's the meniscus level drops, so like this water line drops as the starch is broken down. It's broken down by the amylase that's present in this solution. So amylase and starch are inside the dialysis tubing. So as amylase, this enzyme, breaks down starch, it's going to release maltose, which is small enough to pass through the partially permeable membrane of the dialysis tubing. And as the solute concentration drops, because this starch, this maltose is leaving, it's going to cause like a higher concentration, solute concentration outside. And as we know, the mechanism of osmosis is where water molecules move from a region of high to low water potential. If you have more solutes outside, then that means it has lower water potential, and therefore water is going to move out, and that's how the meniscus level drops from here down here. And then you just know it just moves out. And in this second experiment, they put Benedict's solution inside of this, outside of the dialysis tubing, and the Benedict solution present, that basically illustrates what's going on here outside of the dialysis tubing. So Benedict solution, it tests for reducing sugars. So as the starch is broken down into maltose by amylase, the maltose will leave the tubing via a process of diffusion, so high to low concentration gradient. So this Benedict solution will test for the presence of these sugars, and when it picks that up, it's going to change color, or it's just going to turn red, shown in this diagram, as this maltose is detected. So yeah, that's pretty much it, and thank you for watching this video, and I hope that was helpful.